It's great to be here. Really great that I can tell you a little bit about all-in meta-analysis, which is a new statistical approach to realize the goals of evidence-based research. And this is work that I did while at CWI in the Netherlands, which is the National Research Institute for Mathematics and Computer Science, in collaboration with my colleagues, Professor Peter Grunwald, Dr. Alexander Lee, and Muriel Perez. So at the moment, I'm in a position at the Amsterdam University Medical Centers. So that puts me into a nice place to also put these ideas into clinical practice. And I'm seeing opportunities for that more and more. So all-in meta-analysis. To introduce this idea, I want to take you back to the early days of the COVID-19 pandemic, where we had no COVID-19 specific vaccines, and existing studies, like early studies, were into repurposing existing treatments and vaccines. And one of those early trials in the Netherlands investigated the BCG vaccine, originally developed to protect against tuberculosis, to see whether it could also protect against COVID-19. And this started very early and also inspired many other trials around the world, such as this trial in Australia that was also quite early. And the reason I'm telling you this is that the Dutch BCG researchers were in close connection. They were in close collaboration with all their colleagues around the world. They had shared their protocol, such that many of these other trials were literally inspired by their study design and designed similar trials that could easily be analyzed together in a meta-analysis. Also, they realized that the data would come in slowly. You can vaccinate a large group of participants, but you got data as soon as you see the COVID infections occurring. And especially if lockdowns turn out to be very effective, the wait for the COVID infections for the actual data would be longer. Also, the data would be collected simultaneously around the world. And the statistician on the team, Henry van Werkhoven, saw a worry there. If all these trials are asking the same question, there is a multiple testing issue, that as soon as someone finds a beneficial effect, it poses a difficult decision to the other trials about whether they should continue their trial, whether that would be ethical if there's already a beneficial result. And if maybe at a later stage all those results would come out of all the trials put together in a meta-analysis, and in relation to the others, this first trial would show to be a fluke, an outlier, then it would be difficult to get all those trials back up again. So Henry van Werkhoven already realized this and already talked to all the colleagues around the world about this problem when we came in with the solution. And that solution was to do a meta-analysis that is all in, meaning anytime, live, and leading on interim data from the trials. And I'm putting live and leading in here because the meta-analysis itself can be the leading source of information for any decisions to start, stop, or expand trials. So evidence-based research. And this is what that looked like. We had a dashboard that showed live results. And this one is in demo mode. So this is actually synthetic data. But what we're looking at is a value for the evidence, an e-value for each trial, the yellow to red colors, and in blue, the meta-analysis. And this e-value is a notion of evidence. The larger it is, the larger the evidence in favor of beneficial effect of the vaccine. And as soon as it reaches 400, we can make a decision with a type 1 error guarantee at level alpha 1/400. So 1/400 is 0.2.5% alpha level. And that's the first benefit of this, its simplicity. This meta-evidence simply multiplies the study evidence. So all the evidence in the studies, the e-values, if you multiply them together, you get the blue line. Such that what you see is also the shape of the studies represented in the meta-analysis. For example, this dip, you see the trials contributing to that. Also, because this type 1 error guarantee is continuous for continuous monitoring, you can make all those decisions no matter how you like, and you can reach a conclusion early. So obviously all those trials independently observe much less data than the meta-analysis of all the trials together, such that the meta-analysis reaches this threshold much earlier, and you can conclude the research effort and make decisions based on that. And finally, because it's so simple, this accumulating evidence also allows you to prepare for a future conclusion. So this multiplication of e-values, I plotted it, the y-axis, on a logarithmic scale, such that you see the increase as a linear increase. And linear increases are easy to extrapolate, such that you can guess like how much more time or how many more events you need to reach a conclusion, and can prepare for that, or advise trials based on this expectation of soon reaching the threshold. Live meta-analysis is also exciting. So if you are really in this collaboration while all the trials are ongoing, it makes a connection between trials that also has many other benefits, such as advising each other on trial design. If a new trial comes in, you can discuss all the benefits and of the different designs. You can see what are the best inclusion criteria or definitions for the outcome to nicely fit in the other trials and to ensure homogeneity. You share protocols, make decisions based on design rather than results. For example, do the risk of bias assessment externally and completely unrelated to results. And the sharing of data. So this specific meta-analysis was an IPD, individual patient data meta-analysis, also allows for collaborative data cleaning, which makes it easier to find mistakes and also to do this well because you do it together. So the motivation to work on these statistics was actually already evidence-based research. So I knew about living systematic reviews and the research waste papers, and I saw an interesting challenge there in terms of statistical methodology to make all these intermediate decisions and to create a statistical approach that would still be valid. So even before the pandemic came, our work on e-values was already well suited to this in terms of testing, hypothesis testing as a way to make decisions, but also in terms of estimation, because there is a related approach called anytime valid confidence intervals that share these same characteristics. Also, these intervals, you can monitor them over time. You can make decisions based on them. So if you, for example, decide as a field that you need a 50% vaccine efficacy for this vaccine to be useful, then in this simulated data, you see the point where the 50% vaccine efficacy drops out of the interval. And you can also make a futility conclusion based on that with your anytime valid guarantees. So in presenting this approach, I don't only present the statistics. There's actually other categories of benefits that I usually mention. So statistics, efficiency, collaboration, and communication. The statistics is a really real step forward, because in a situation of continuous updating, which we would like to do also in living systematic reviews, it's actually quite difficult with other statistical approaches to retain type 1 error guarantees. And the other sequential methods that are out there are actually advised against by Cochrane, for example, to use them in living systematic reviews. Efficiency. There are other problems in the way we put statistics to practice. When we do a power analysis, we're actually calculating the probability of an all or nothing decision. And when a trial is not able to reach their target, their own recruitment target, we just call it underpowered, and we don't value it as much. Well, it's really only this idea of doing it with one study alone, one study and then an all or nothing decision, that makes a study underpowered. You could also see it as a brick building towards a building of evidence. Collaboration. Doing this on interim trial results is exciting, and it also allows for other improvements to the research process, such as preventing publication bias. If you're in this from the protocol phase, it's really weird that you filed your results. And finally, communication. There's also a debate in statistics how to resolve, for example, the replicability crisis and how statistical methodology hurts science. And there's many different flavors of what statisticians like as interpretation of statistical results. E-values position themselves very nicely within that debate because they have more than one interpretation. You can think of them as likelihood ratios, and they're often very closely related to likelihood ratios. Fortunately, there's hardly any statistician that doesn't like likelihood ratios. They can be inverse p-values. If you like p smaller than alpha as an interpretation, you can just think of E larger than 1 over alpha as the same thing. You can think of them as base factors. If you like the Bayesian view, if you interpret an E-value as a base factor, you're also fine. And most of all, the one that I really like is they are betting scores. And that connects them to the accumulation of evidence. Betting scores in the sense that we also have investment portfolios, that money can grow under uncertain circumstances. These things also relate to how evidence accumulates in science, the way money can accumulate. And you can think of E-values as money in the same way. So the future of evidence-based research, is it all in? I hope so. I think it helps to answer these questions, like how to decide when would you not need more research. If you can agree on a threshold with your field, for example, if you can agree on 100 as a threshold with a 1% type whatever rate, you can just use that threshold and compare your E-values to that threshold under any evidence-based research decision. So under any data-driven decision on doing more studies or less studies. While such study decisions actually introduce meta-optional stopping, introduce bias in conventional meta-analysis and in Bayesian approaches. Other question, how to design a new study in the light of existing evidence. If the existing evidence already gives 10 and you've decided on this threshold of 100, it's pretty easy that you need to design to reach another 10, because 10 multiplied by 10 makes 100. And you can take that as the approach. So no power analysis on your study alone, but really power analysis on reaching the conclusion of the field as a whole. Funders can ask for such power analysis to add what is already there, even look at registries, at protocols of ongoing trials, and see what are those trials aiming for in terms of E-values. And if you know that that's what they are doing, how large does your study need to be to add to that? And if you can get your hands on it, interim E-values are just as easy to handle as E-values of completed trials. So that brings me to the end. Here are some references. I would also really like to point you to my PhD thesis. That is really in the spirit of this betting approach to the interpretation of E-values. You can guess now where the all-in acronym comes from. It also has an introduction chapter that introduces evidence-based research and its connections to E-values and the interpretation of accumulating money as accumulating evidence in a way for non-statisticians. So with that, thank you very much.